Now that we've learned about functional groups and physical properties of organic compounds, as well as the intermolecular forces responsible for those physical properties, we're going to turn our attention to biomolecules. Our first group of biomolecules will be carbohydrates. Here are a few examples of carbohydrates or sugars. They have either an aldehyde or a ketone and lots of alcohols. This is glucose, important in many biochemical pathways, fructose or fruit sugar, and ribose, which is the sugar in RNA. Here we have different representations of glucose. Here we are showing a little bit about the stereochemistry, the exact arrangement of the groups. Actually, we're doing that here as well. It's just using a Fisher projection, which we'll learn about later. But in addition to existing in this linear form, carbohydrates can exist in a cyclic form. It can be either a 5-membered ring or a 6-membered ring. It's the aldehyde or the ketone that are involved in forming the ring, and so notice we don't have our aldehyde or ketone anymore. Instead, this was the carbonyl carbon, and it's now forming a different type of functional group called a hemiacetal. Because of the polarity of glucose, because we have all of these alcohols, sugars are able to hydrogen bond, so they're very soluble in water. In addition to existing in linear and cyclic forms, carbohydrates can also exist as 2 sugars or disaccharides, or many sugars, polysaccharides. Not all the details are shown here, but this is starch and this is cellulose. So monosaccharides, disaccharides, oligosaccharides, which is several sugars, polysaccharides, these are all very important biomolecules which have many important roles in the body. Many sugars play an important role on the cell surface. Sugars can be connected to the cell surface where they act as labels or identifiers. For example, with blood groups, if you have type A, type B, type O, type AB, what you have is different sugars connected to the cell surface of your red blood cells. Now they all have N-acetyl D-glucosamine, followed by D-galactose, followed by L-fucose, but there's a difference with other sugars connected as well. Because carbohydrates have so many OH groups, that means they can engage in hydrogen bonding. And so they can make very strong intermolecular forces between molecules, as well as strong intermolecular forces with polar solvents, such as water. Another group of biomolecules would be the amino acids. So this is the general structure of the amino acids. They are alpha beta amino acids. Often in chemistry, we will use Greek letters to be able to indicate relative locations. And so starting with the carboxylic acid carbon, the next atom is called the alpha position, the next is called the beta position, gamma, delta, etc. Now, amino acids, we have the carboxylic acid and we have the amine. There's only one carbon in the middle, but then they vary in the identity of this R group. If R is just hydrogen, this is glycine, a methyl group, alanine, a 3-carbon group, isopropyl group, this is valine. We have aromatic rings, and additional rings. We have amides, as well as alcohols. We have thiols, more carboxylic acids or carboxylates, and more amines or ammonium ions. The amino acid itself is involved in the long chain of amino acids that make up a protein. And then the side group, the R group, imparts different properties as the protein packages itself up. With all of these different functional groups, we can see that different amino acids are capable of different types of intermolecular forces. Different intermolecular forces are really important when we think about peptides and proteins. So here we have a small peptide made up of 4 amino acids. Here is the amine, and then this was the carboxylic acid, but now it forms an amide linkage. This is the amine. This was the carboxylic acid, is now the amide, the amine, now the amide, the amine, and the carboxylic acid. As an organic chemist, you would call this an amide, but because it's in a protein, this is called a peptide bond. Peptides and proteins, which are huge chains of amino acids, have primary, secondary, tertiary, and quaternary structure. Primary structure refers to the exact order and identity of the different amino acids. So if this first one is a glycine, this is a hydrogen, and the second one is an alanine, where this is a CH3 group. Exactly that order is given by the primary structure. The secondary structure is based on interactions between the amine and the carbonyl in adjacent chains. So here we have hydrogen bonding between an NH and a carbonyl in a neighboring chain. And so secondary structure is determined by main chain interactions. And secondary structure gives you things like an alpha helix or beta sheets. Tertiary structure is based on interactions between the amino acid side chains. So there are certain side chains that are nonpolar, and so those will interact with each other by induced dipole or dispersion interactions. There are some amino acid side chains that are positively charged and negatively charged, and so those can form something like an ionic bond called electrostatic interactions, or it can be called a salt bridge. Other amino acids that have an OH or an NH in their side chain, they can be involved in hydrogen bonding. And so this shows the structure of a particular enzyme, and there are some regions that have the alpha helices, as well as these yellow arrows. These are the beta sheets, and then the side chains will interact with each other, either with hydrogen bonding or induced dipole interactions or electrostatic interactions. Typically, on the outside of the protein, this is where we're going to have many amino acids that can hydrogen bond with water because these proteins are in an aqueous environment. And then the interior of the enzyme, that might have a lot of the hydrophobic interactions. So the induced dipole, those types of interactions are going to be kind of in the interior of the protein. So I think with amino acids, peptides, proteins, this is where the importance of intermolecular forces is really evident. The next group of biomolecules are the lipids, and lipids actually do not have common structural features, but they are similar in their physical properties because they are all less soluble in water, more soluble in organic solvents. Because for the most part, lipids are very nonpolar and they can only interact by induced dipole interactions or dispersion forces. Now, many lipids are based on fatty acids and fatty acid derivatives. So here we have a fatty acid. This is another fatty acid. Even though we have this carboxylic acid that's capable of hydrogen bonding, because we have so much of this molecule that's nonpolar, then overall this can only do induced dipole interactions. This is an example of a saturated fatty acid. This is an unsaturated fatty acid, and naturally occurring unsaturated fatty acids are always cis, so that both alkyl groups come off the same side of the alkene. You've also heard of trans fats. That's different than the naturally occurring fatty acids. In addition to fatty acids themselves, there are many other important molecules that are derivatives of fatty acids. This one is a wax. So notice this is an ester of a fatty acid. This might be in a waterproof coating on a leaf, for example. Here we have a triester with glycerol. So glycerol is a trialcohol, a triol, and then we have fatty acids, and these form esters. So this is an example of a triacyl glyceride. you're too young to worry about this yet, but as you get older, your doctor will do blood tests to check your cholesterol levels and your triacylglyceride levels. These are very important in energy storage. Here, this is again glycerol, and then we have a couple of fatty acids, and then we have this other group. This is a phospholipid. Phospholipids make up cell membranes. Now, you've probably seen this represented as a whole bunch of little squiggles, where this is the hydrophilic head and then the hydrophobic tail. Well, this is the hydrophilic head, and then the fatty acids make up the hydrophobic tail that makes up the lipid bilayer of a cell membrane. These are much more complex, but they're still based on fatty acids. This is a prostaglandin, which is a chemical messenger. And this is prostacyclin, another chemical messenger that inhibits platelet activation. This serves to counter thromboxane, which is yet another lipid chemical messenger that promotes platelet aggregation, and then prostacyclin prevents it so that the body keeps the correct balance of forming platelets, forming clots when there's an injury, but not forming clots in general. In addition to the fatty acid derivatives, we have steroids such as cholesterol. And notice we have this six-membered ring, six-membered ring, six-membered ring, five-membered ring. So that's the common skeleton in all the steroids. And the steroids include the male sex hormones, testosterone and androsterone, as well as the female sex hormones, progesterone and estradiol. Another group of lipids would be the terpenes. These are all biosynthesized in the same way, at least in the early steps, even though there is a wide variety of structures. Citronellal, this is an aldehyde and it has an alkene, and this is what we have in citronella candles that help keep mosquitoes away. Geraniol, we have an alcohol and we have a couple of alkenes. This is in roses. Menthol has a couple of OH groups. This is what we have in Vicks VapoRub, for example. Here we have carvone. We have a ketone, an alkene, and another alkene. This is responsible for the odor of spearmint as well as caraway seed. So really different. This is vitamin A, what we need to help our vision. And then this is caryophyllene. I like to think of this as a little Pac-Man molecule. This can be isolated from the essential oils of cloves and many other spices. Now, lipids are really quite interesting because in general, I think they get a bad rap because you don't want to have fats. But lipids in general have a wide variety of important functions. So they're responsible for energy storage, they're the component of cell membranes, they insulate nerve cells, they're vitamins, water repellent, protective coatings, insect repellents, hormones, chemical messengers. So all these different functions. And they're involved, either they influence or regulate heart rate, platelet aggregation and clotting, fertility and conception, metabolism, constriction of bronchial passages. So if you have asthma, you can thank a lipid for that, as well as electrolyte balance. All of these lipids are going to be very water insoluble, much more soluble in organic solvents. On the previous slide, there was a phospholipid, and so that makes up cell membranes, but steroids also exist in between the phospholipids. So this is very nonpolar. It exists within the hydrophobic region, the nonpolar region of the phospholipids, and this helps to give a little bit of structure so that the cell membranes aren't just super floppy. So although lipids have very different structures, they are all very nonpolar, capable primarily of induced dipole or dispersion forces as their intermolecular forces, but they still have many, many, many important roles. And finally, the last group of biomolecules we're going to talk about are the nucleic acids. So this includes DNA and RNA. So here we have some deoxyribonucleotides. So this is the deoxyribose sugar, which is connected to a phosphate, and it's connected to a nitrogen base, in particular adenine. This one has the sugar phosphate nitrogen base guanine, sugar phosphate nitrogen base cytosine, sugar phosphate nitrogen base thymine. The ribonucleotides are similar except that there is another OH group on the sugar because this sugar is ribose and this sugar is deoxyribose. So it's missing an OH group. The same, we have adenine in both cases, we have guanine in both cases, we have cytosine, but here's the difference. We have thymine in DNA, but uracil in RNA. Now, the way that amino acids come together, we have our phosphate connected to our sugar is connected to the next phosphate's connected to the sugar, and so on. So we have the 5' end, and then down to the 3' end, where we have phosphate sugar, and over here's the base, phosphate sugar, over here is the base. So we have alcohols that are capable of hydrogen bonding. We have very polar phosphate groups. And then we have these nitrogen bases, which are all capable of hydrogen bonding. The hydrogen bonding between the amine bases is really important. In particular, adenine and thymine hydrogen bond very effectively. Guanine and cytosine hydrogen bond very effectively. And this hydrogen bonding is what's responsible for the structure of DNA. So the sugar phosphate, that makes up the helix. And so you can think of this as the outside of the ladder. And then the amine bases make up the steps of the ladder. So these are reaching across, hydrogen bonding together, and that makes up each rung of the ladder. So this hydrogen bonding, this particular structure of the nucleotides together, this is responsible for the double helix structure of DNA, but it's also responsible for the replication of DNA. So the double helix can unzip, and then each strand of the original DNA is going to form new interactions with new nucleotides, and so new sets of hydrogen bonding between the bases. So this strand can form a new double strand, and this strand can form a new double strand, so we end up with twice the amount of DNA, and it's a perfect copy. So the things that we're learning about functional groups and intramolecular forces applied to organic molecules are also incredibly relevant when it comes to biomolecules and with the different types of interactions that are responsible for all the biochemical processes that happen in nature.